This is part 1 for the reproductive system lecture, part 1 of 3. This lecture will deal primarily with the male reproductive system. So the sexual reproduction is going to be simply a process in which organisms are going to produce offspring by means of germ cells that are called gametes. The organs of reproduction are going to be grouped in 3 categories. We've got the gonads, the ducts, and the accessory glands. The gonads are going to be responsible for producing gametes and secreting hormones. The ducts are going to transport, receive, and store those gametes. And then the accessory sex glands are going to produce material that's responsible or that's needed to support gametes. Functionally, we're talking about the male reproductive system here. There are going to be 4 main functions for the male reproductive system. The testes are going to produce sperm as well as the male sex hormone, those androgens. The ducts are going to transport, store, and assist in the maturation of sperm. The accessory sex glands are going to secrete most of the fluid portion of the semen, and then the penis is going to contain the urethra, which is a passageway for ejection of semen as well as the excretion of urine. So the organs of the male reproductive system are going to include the testes, a system of ducts which includes the epididymis, the ductus deferens, the ejaculatory ducts, as well as the urethra. The accessory sex glands are going to include the seminal vesicles, the prostate, and the bulbourethral glands. And then there are going to be several supporting structures, which are going to include the scrotum and the penis. So the testes, these are the male gonads, are responsible for producing sperm and secreting hormones. The duct system that transports and stores sperm, as well as assists in maturation, assisting in their maturation, as well as conveying them to the exterior, to the outside world. And then semen is going to contain sperm, plus some of the secretions that are provided by those accessory sex glands. The supporting structures are going to have various different functions. So the penis is going to deliver sperm into the female reproductive system, and the scrotum is going to support the testes. The scrotum is going to be a sac of loose skin and underlying subcutaneous tissue that's going to contain the testes. This is also known in the common terms as the ball sac. Internally, the scrotum is going to be separated into 2 compartments by the dartos muscle, as well as a subcutaneous layer. And the reproduction and survival of the spermatozoa, those little initial sperm cells, is going to require a temperature that's lower than your normal core body temperature. And this is why the testicles, are going to kind of reside outside of the body cavity. So the temperature of the testes is going to be regulated by the cremaster muscle. This is going to elevate them and bring them closer to the pelvic cavity when it's cold, and it's also going to relax and cause the testes to move farther from the pelvic cavity when it's hot outside. So this is going to sort of help to keep them in an appropriate temperature range so that those spermatozoa can develop properly into fully formed little sperm. The testes are going to be a paired, sort of oval-shaped gland. It's going to reside in the scrotum, and it's going to be partially covered by the tunica vaginalis. The internal to that tunica vaginalis is a connective tissue capsule called the tunica albuginea, and the tunica albuginea is going to extend inward from the septa that divides the testes into a series of internal compartments called lobules. All right, so we have, let's see where to go, our lobules here that are produced by these little extending inward bits of the tunica albuginea. All right, each lobe is going to contain 1 to 3 tightly coiled tubes called seminiferous tubes that you can see over here, right here, the seminiferous tubes. And this is where sperm is produced. This whole process of producing sperm is called spermatogenesis. So these seminiferous tubes are going to carry sperm that's produced within them out of the testes. The seminiferous tubules are going to contain two types of cells. We've got our spermatogenic cells. These are stem cells. These are our sperm-forming stem cells. And then we have our sustentacular cells, which you can see here. These are going to have several different functions that are going to be mainly in supporting spermatogenesis. We'll talk about those in just a little bit. Stem cells called spermatogonia are going to develop from primordial germ cells that are going to arise from the yolk sac and are going to enter the testes during the 5th week of development. So it's starting fairly early, this kind of preparation to develop sperm. In the embryonic testes, the primordial germ cells are going to differentiate into spermatogonia, which are going to remain dormant during childhood, and then they are going to actively begin to produce sperm at puberty. So towards the lumen of these seminiferous tubules are going to be layers of progressively more mature cells. After a sperm cell has formed, it is released into the lumen of that seminiferous tubule. A little too far. That leads us into our sustentacular cells, which we mentioned before. So embedded around those spermatogenic cells in the tubules are going to be sustentacular cells. These are going to have tight junctions between these two cells, or between these sustentacular cells, that are going to form the blood-testes barrier. And this is going to prevent any kind of immune response from being mounted by the immune system that would attack surface or recognize surface antigens on those spermatogenic cells and target them for identify them as something foreign. So the sustentacular cells are also going to be involved in nourishing those spermatocytes, the spermatids, and those spermatozoa, all of the different degrees of maturity for sperm cells there. They're also going to mediate the effects of testosterone and follicle-stimulating hormone on spermatogenesis. They're going to be involved in phagocytosing any kind of excess spermatid cytoplasm as the whole developmental process occurs. And we'll look more at that in just a minute. And they also are involved in controlling movements of our spermatogenic cells and the release of our spermatozoa into the lumen of the seminiferous tubules. And then lastly, they're going to secrete fluid for the sperm to transport, as well as they're also going to secrete the hormone inhibin. So the other cells that we can find in the testes, there are a couple of other cells that we can find in the testes. In the space between adjacent seminiferous tubules, there's going to be clusters of cells called interstitial cells, or also called Leydig cells. These cells are going to secrete testosterone, and this is the most prevalent androgen in men. So an androgen, just as a brief reminder, is a hormone that promotes the development of any sort of masculine characteristics. Anything from deep voices to extra body hair to larger, potentially larger muscle mass or denser skeletal systems. But anyhow, so testosterone is the main androgen that is present in males, and this testosterone is also going to promote a man's libido or sex drive. So here's just a slide for your own sort of just information there, looking at a review of the reproductive cell division of the various stages of meiosis, which is going to be important for some of our later discussions. Specifically, we need to know about meiosis for the process to understand better the process of spermatogenesis. This process of spermatogenesis is going to be the process by which the seminiferous tubules of the testes are going to produce haploid sperm. So they only have half the number of chromosomes that you would find in a diploid sperm, or a diploid cell. So, in the first step, it begins with our diploid spermatogonia. Up here we've got our stem cell. This is going to undergo mitosis, so that way we're going to have two of our spermatogonium, so we're going to have two of these. One that can be saved for later so that we can continue to have sperm cell production, and then one that's going to differentiate. So after this mitosis occurs, we have our reserve future stem cell, and we're also going to develop cell, as well as a cell that can be developed into a secondary spermatocyte for sperm production. So in step 2, the diploid, this is going to differentiate into a primary spermatocyte, and this primary spermatocyte is going to undergo meiosis 1 to form haploid secondary spermatocytes, which you can see here. Each of these has one copy of the chromosome, of the genome, instead of 2. So in step 3, meiosis 2 is going to result in the formation of the haploid spermatids, which we see here. And the spermatids are all going to be connected by cytoplasmic bridges. The final stage of spermatogenesis is called spermiogenesis, and this is when we have the maturation of the spermatids into sperm cells or spermatozoa. And this is the release of these from the release of a sperm from its connection to those sustentacular cells is going to be known as spermiation. Right. So, looking at a little bit of sperm anatomy, the sperm is going to basically be designed to reach and penetrate the secondary oocyte in order to achieve fertilization and create a zygote. The head of the sperm is going to contain a nucleus with 23 chromosomes, and then an acrosome, which we see up here, which is like a little hat for the head of the sperm. This is going to cover the head of the sperm, and it's going to contain enzymes that are going to help with penetration of that oocyte so that fertilization can occur. The neck of the sperm is going to contain the centrioles that are responsible for forming the microtubules that are going to make up the rest of that tail. And then the middle piece, which we can see here, is going to contain the mitochondria that are necessary for the production of ATP. So that basically they're producing the ATP that is going to provide energy for locomotion or movement of that sperm. And then last but not least, we have our principal piece. And our end piece. This is going to be the really functional part of the tail. It's actually going to do like the flapping back and forth for the actual swimming or movement of our sperm. These sperms are going to be produced at a rate of about 300 million per day, and once ejaculated, they're going to have a life expectancy of about 48 hours within the female reproductive system. And their function is going to be primarily to fertilize those secondary oocytes. So, looking at the hormones that are going to be responsible for controlling this sperm production and release, there are going to be several different hormones that control testicular function. So, at puberty, gonadotropin-releasing hormone is going to stimulate the cells that are in the anterior pituitary gland to produce luteinizing hormone and follicle-stimulating hormone. Luteinizing hormone is going to be responsible for stimulating cells in the testes to produce testosterone, and then follicle-stimulating hormone is going to stimulate spermatogenesis, so the development of these little sperm cells. So in some target cells, such as the external genitalia, as well as the prostate, the enzyme 5-alpha reductase is going to convert testosterone into another androgen called dihydrotestosterone, which we'll talk about a little bit more later. But this is an important one for several different things, including the male pattern of development for before birth. So what makes you grow a penis and testicles, etc. As an embryo. It's also important for the enlargement of male sex organs and stuff. So it does quite a few things, but we'll talk about that a little bit more later. So looking at hormonal control, this is very much like a feedback loop where follicle-stimulating hormone is going to act indirectly to stimulate spermatogenesis. Follicle-stimulating hormone as well as testosterone are going to act synergistically on those sustentacular cells to stimulate the secretion of androgen-binding proteins, ABP. These secretions into the lumen of the seminiferous tubules are going, and in the interstitial fluid around those spermatogenic cells. This is going to help with, So ABP is then going to bind to testosterone, and this is going to help to keep that concentration high, so it can't get broken down and secreted and filtered out by the kidneys and the liver. The testosterone is going to stimulate that final steps of spermatogenesis that are going to take place in our seminiferous tubules. And then once the degree of spermatogenesis that's required for the male reproductive function has been achieved, those sustentacular cells are going to release a hormone called inhibin, and this is going to inhibit the follicle-stimulating hormone secretions by the anterior pituitary. If spermatogenesis is proceeding at a rate that's a little too slow, then less inhibin is going to be released, which is going to allow for more follicle-stimulating hormone to be secreted, and in turn this is going to lead to an increase in the rate of spermatogenesis. So, coming back to the role of testosterone and other androgens in the body, all right? These are going to be very important for a variety of different things. So as far as prenatal development, before birth, testosterone is going to help to stimulate the male pattern of development of the reproductive system ducts, as well as the descent of those testes. Dihydrogen testosterone is also going to stimulate the development of external genitalia, so prenatally, like when you're in utero still. And testosterone is also going to be converted in the brain into an estrogens. This is one of the feminizing hormones. And this is going to play a role in, we think this plays a role in the development of certain regions of the brain in males. Then another big role of testosterone and androgens in the body is the development of those male sexual characteristics. So at puberty, testosterone and dihydrotestosterone are going to bring about the development and the enlargement of the male sexual organs, as well as the development of masculine secondary sexual characteristics. This is things like increased height, increased bone mass, the deeper voice, the increase in hair growth in other places, the secondary sexual characteristics, things that are not directly related to reproduction, but are generally associated with being male or female. Like a larger Adam's apple for male secondary sexual characteristics. Breasts are a secondary sexual characteristic for females. The secondary sex characteristics are basically going to be traits that are going to help to distinguish males and females, but like I said, don't play any direct role in reproduction. The testosterone and other androgens are also going to be very important for the development of sexual function. So androgens are going to contribute to male sexual behavior, as well as spermatogenesis, as well as sex drive or libido in both males and females. So you should remember from previous lectures that the adrenal cortex is going to be the main source of androgens in females, since obviously they don't have testicles to be producing this testosterone. And then last but not least, testosterone and other androgens are going to be very important for the stimulation of anabolism, which, if you remember from previous lectures, is the building up of larger molecules from smaller pieces. So androgens are anabolic hormones, and that is they're going to stimulate protein synthesis, so the building up of larger proteins from smaller bits. This effect of the stimulation of anabolism is going to be obvious in the heavier muscles, as well as the increased or larger bone mass that you'd see in most men in comparison to women. So, as with most things in the body, there's going to be a negative feedback that's going to regulate testosterone production. Like we've mentioned before, negative feedback loops are the most common kind of feedback loop. So when testosterone concentrations in the blood are going to increase past a certain level, it inhibits the release of gonadotropin-releasing hormone by cells in the hypothalamus. As a result, there's going to be less gonadotropic releasing hormone in the portal blood that's flowing from the hypothalamus into the anterior pituitary. This causes the gonadotropes in the anterior pituitary to release less luteinizing hormone, so that the concentration of luteinizing hormone in the systemic blood is going to drop. This drop in luteinizing hormone in the circulating blood is going to result in interstitial cells in the testes secreting less testosterone, and there's also going to be then a return to homeostasis. If the testosterone concentration in the blood falls too low, however, our gonadotropin releasing hormone is again going to be released by the hypothalamus, and it's going to stimulate the secretion of luteinizing hormone by the anterior pituitary. And then this is going to allow for luteinizing hormone to, in turn, stimulate testosterone production by the testes. So, let's look a little bit more closely at the actual anatomy of the testes, or the testicles. For starters, we're going to look at the ducts of the testicles. So pressure is going to be generated by the fluid that's secreted by the sustentacular cells. This is going to push sperm and fluid along the lumen of seminiferous tubules and then into a series of very short ducts called straight tubules. You can see these ones here. Those are our straight tubules. Boop. These straight tubules are going to lead to a network of ducts in the testes called the Redes testes, which is right in here. And then from the Redes testes, sperm is going to move into a series of coils called the efferent ducts, which you see here. And these are located in the epididymis that are going to empty into a single tube called the ductus epididymis. The epididymis is going to be a comma-shaped kind of organ that you can see here. This is layer. And it's going to lie along the posterior border of the testes and is going to be the site of sperm maturation. The sperms are going to be transported out of the testicles through the efferent tubes into the epididymis, which is then going to empty into a single tube called the ductus epididymis. So this ductus epididymis is going to be lined by stereocilia, and this is going to be the final site of sperm maturation, so where they just finish their maturation process, as well as storage. So sperm may be stored, may remain in storage here in our ductus epididymis for at least a month, after which they're either going to be expelled or they're going to degenerate and be reabsorbed into the testicles. So the next part of the male reproductive system for the ducts of the testicles is the ductus deferens, or the vas deferens, and this is going to exit the tail of the epididymis, and it's going to ascend through the spermatic cord and into the pelvis. All right, then it's going to loop over the ureter, which we see here, and then it's going to pass over the side and down the posterior surface of the urinary bladder, which we can see here. So this is the dilated terminal portion of the ductus deferens, or the vas deferens, and this is called the ampulla. So our vas deferens is going to be responsible for storing sperm and then propelling them towards the urethra during ejaculation. Next is the spermatic cord. This is going to ascend out of the scrotum, and it's going to contain the ductus deferens, the testicular arteries, the veins that are responsible for draining the testes, as well as the autonomic nerves, lymphatic vessels, and the cremaster muscles that we mentioned before. The spermatic cord and the ilioingual nerve are going to pass through the inguinal canal, which is going to originate at the deep inguinal ring. This is basically just a slit-like opening in the aponeurosis of that transverse abdominal muscle, and it's going to end at the superficial inguinal ring. The principal method of sterilization for males is called a vasectomy, and this is when both of the ductus deferens are going to be cut and tied, and this results in no sperm entering the ejaculate. So you can still ejaculate, but no sperm are going to be present in that ejaculatory fluid, or in the semen. Next, in the male reproductive system is the ejaculatory duct. This is going to arise from the junction of the duct from the seminal vesicles and the ampulla of the ductus deferens. So their function is going to be to eject the spermatozoa into the prostatic urethra. The urethra is the duct that's shared by the reproductive and the urinary system. So this is where both semen and urine are going to pass through the urethra. And the urethra is going to pass through the prostate gland. This is our prostatic urethra, through the deep muscles of the perineum, as well as the penis. So, looking now at the accessory glands of the male reproductive system. We've got our seminal vesicles, the prostate, and the bulbourethral or the Cowper's gland. So we'll talk about each of these individually just in a minute. So, for starters, we've got the seminal vesicles. These are going to secrete an alkaline, sort of viscous fluid that's also going to contain fructose, prostaglandins, and clotting proteins. The alkaline nature of the fluid is going to help to neutralize the acid that's in the male urethra as well as in the female reproductive tract. The fructose is going to serve as an energy source for the production of ATP by those sperms, so kind of a little snack for the road, if you will. And then prostaglandins are going to contribute to sperm motility and viability. So seminogelins is going to be a protein, the main protein in semen that's going, or seminal secretions, that's going to cause coagulation of semen after ejaculation. And it's thought that this coagulation is going to occur in order to help to keep those little sperm cells from leaking from the vagina. So the fluid that's secreted by these seminal vesicles is normally going to constitute about 60% of the volume of semen. So the majority of semen, of the volume of semen, is coming from these seminal vesicles. 60%, more than half. The next accessory glands we have are the prostate gland. This is responsible for secreting a milky, kind of slightly acidic fluid that's going to contain citric acid, which can also be used by the sperms for ATP production. It'll also have acid phosphates, as well as several proteolytic enzymes, such as prostate-specific antigens, or PSAs, as well as a couple of other types of enzymes that are going to be there to help to liquefy coagulated semen. So it can kind of become more liquid again and move to wherever it is supposed to go. The bulbourethral glands are going to secrete mucus and are also going to that is important for lubrication, and it's also going to secrete an alkaline substance that's going to help to neutralize acid. So those are our three accessory glands for the male reproductive system. Next, we have semen. Semen is also called seminal fluid. This is a mixture of spermatozoa as well as accessory sex gland secretions that's going to help to provide the fluids in which those spermatozoa are going to be transported. It also provides nutrients, and it neutralizes any acidity of the male urethra and the female vagina. So semen is going to contain an antibiotic as well as seminal plasmin, as well as a prostatic enzyme that's going to help to coagulate and then liquefy semen in order to aid its movement through the uterine cervix. Once it's been ejaculated, liquid semen is going to coagulate within about 5 minutes due to the presence of clotting proteins in that that's coming from those seminal fluids, from that seminal vesicles. Then after about 10 to 20 minutes later, after it's coagulated or clotted, semen is going to reliquefy because the prostate-specific antigen is going to get in there and help to reliquefy reliquefy those semen to help to break down that clot. So, moving on then to the penis. This is one of our accessory sexual structures. This penis is going to contain the urethra, and this is going to be a passageway for the ejaculation of semen, as well as the excretion of urine from the body. The penis is cylindrical in shape, and is going to consist of a body, the glans penis, and the root. So the body of the penis is composed of 3 cylindrical masses of tissue, each of which are surrounded by fibrous tissues that are called the tunica albuginea. There are going to be 2 dorsolateral masses that are going to be on either side, which you can see here. These are called the corpo cavernosa penis on either side. You can see this is going to make up the majority of it. And then there's a smaller sort of mid-ventral mass called the corpus spongiosum penis. And this is going to contain the spongy urethra, and it's going to help to keep it open during ejaculation. The skin and the subcutaneous layers of the penis are going to be enclosed by all 3 of these masses. And these parts are going to be consisting of erectile tissue. So this erectile tissue is going to be composed of numerous blood sinuses. This is just vascular spaces that can fill with blood, essentially. And these vascular sinuses, are lined by endothelial cells and are surrounded by smooth muscle as well as elastic connective tissue. This is what is responsible for filling with blood and causing an erection to occur, making the penis hard or standing upright. The distal end of the corpus spongiosum penis is going to be slightly enlarged, and there's going to be a sort of acorn-shaped region, which I guess you can see there. It's kind of a weird, not really what I would call acorn-shaped, but who might argue. This is called the glans penis, and this is going to be, it's going to have its margin is going to be the corona, which means crown. So this is the crown of your penis. The distal urethra is going to be enlarged within the glans penis, and this is going to form a terminal slit-like opening that's called the external urethral orifice. And then covering the glans penis of an uncircumcised penis is going to be a loosely fitting kind of flap of skin called a prepuce or a foreskin. So it used to be pretty common practice to have this removed in infancy. Still is done for various religious reasons. This whole practice would be called a circumcision, where the foreskin of the penis would be just simply removed. Some people say it's better for cleanliness. Some people say it doesn't make any difference. It really doesn't seem to matter either way. But some people do it for religious reasons as well. This removal of this foreskin. So, next, the penis is also going to have a root. So the root of the penis is the proximal portion. It's the part that's closest to your actual body. The root of the penis is going to consist of the bulb of the penis, the expanded posterior, which you can see here, the bulb of the penis, the expanded posterior continuation of the base of the corpus spongiosum penis, and then the crura of the penis. And this is going to be 2 separated and tapered portions, which you can see here, of the penis. These are tapered portions of the corpora cavernosa penis. So the bulb of the penis is going to attach to the inferior surface of the deep muscle of the perineum, and is also going to be enclosed by the bulbospongiosum muscle. So this is a muscle that's going to aid in ejaculation. Then each of the crus of the penis are going to is going to bend laterally away from the bulb of the penis, which you can see here, laterally away from the penis. And this is going to attach to the ischial and the inferior pubic rami, and this is going to help to this is going to be supported by the ischiocavernosus muscle. So the weight of the penis is going to be supported by 2 main ligaments that are going to be continuous with the fascia of the penis. We have the fundiform ligament. This is going to arise from the inferior part of the linea alba, and then the superior ligament of the penis, which is going to arise from the pubic symphysis. So Last but not least, we have erections, looking at the neurological implications of an erection. So an erection is going to be brought on by parasympathetic innervations that are going to lead to vasodilation of arterioles in erectile tissue. So large amounts of blood are going to enter the tissue, into those dilated blood sinuses, and then ejaculation is going to be a powerful release of semen due to sympathetic stimulation. So the bulbospongiosus, the ischiocavernosus, and the superficial transverse perineal muscles are all going to contract in order to help to force that semen out of the penis. And that is all that we have for the part 1 of the reproductive lecture. So as always, please email me if you have any questions, comments, concerns.